Hi everyone, my name is Maddie and I'm doing my presentation on trichomoniasis. Alright, so first we're going to start off with the microbe that causes this disease. So the microbe is Trichomonas vaginalis. So this is an anaerobic organism, which means that it thrives in environments with low to no oxygen. And it's also nice for T. vaginalis because it has adopted to function in anaerobic conditions, specifically in the urogenital tract, which is where this microbe is usually found. It is a protozoan. It has flagella, typically about 4 to 5, and these flagella play a crucial role in the movement of the parasite and help it navigate through the urogenital tract. This microbe or parasite has a pretty unique feature, which is the hydrogenosome. So instead of the mitochondria, it has this hydrogenosome, which is a specialized organelle that is involved in energy metabolism and is capable for producing molecular hydrogen as a byproduct. It is also capable of phagocytosis, so it can engulf particles, including bacteria and the host cells that it attaches to, and it also can absorb nutrients from the environment. And lastly, it reproduces via binary fission. So one cell divides into 2 identical daughter cells. And these pictures are just showing the structure of this microbe. So with T. vaginalis, there are different cellular forms. So the first one is a trophozoite, which is free-swimming, has flagella, pear-like cell shape, and is also the infective form. And you can see in the pictures here that are describing the shapes. The second one is an ambioid, which is a pancake shape, which helps increase surface contact. And there are pseudocysts, which can be induced in the vitro upon exposure of trophozoites to cold and other stressors. So it can change into these cellular forms depending on where it is in the body or the stressors that are involved. Alright, so now we're going to talk a little bit more about trichomoniasis. So it is the most common non-viral STI. It has an undulating membrane. Generally, it is a amoeboid shape when it's attaching to the cells in the vagina, which I mentioned in the previous slide, will help increase the surface area. And then the microbe that causes this disease is commonly found in normal microbiota. So that's pretty interesting as well. Alright, so now we're going to talk about how trichomoniasis spreads. So it is a sexually transmitted infection that is caused by a parasite, usually when individuals are having unprotected genital contact or sex, so it can be vaginal, anal, or oral. This also can be spread from a mother to her infant when childbirth is occurring, so it can be passed from the mother to baby as well. Also, there are asymptomatic carriers, so a lot of times individuals who have trichomoniasis will not always show symptoms, but they can still transmit the infection to their partner or to someone who they are having sex with. So you might not know you have it until you transmit it to someone and then they start showing symptoms. In this picture here, it's just showing how the one of the cellular forms, which is a trophozoite, is in the vaginal and the prostatic secretions, and then it can multiply earlier, like I said, by binary fission, and then the trophozoite will be in the infective stage, and then that's when it will be passed to someone else via sexual intercourse, most likely. So some symptoms, so like I said, Many individuals will be asymptomatic, so they will not show any symptoms. But when symptoms occur, these are some of the symptoms. So for women, it could be dysuria or itching, burning, redness, soreness of the genitalia, and vaginal discharge, usually foul smelling and kind of iffy on the color. Sometimes it could be more like a yellowish color, or it could be white, but it's just going to have a lot more discharge than normal as well. And then for men, there could be itching or irritation inside the penis, and then burning with urination or after ejaculation. And there's also a little bit of discharge coming from the penis of men. So the diagnosis will usually start out with an examination of the genitals. And then there's lab tests that can be done. So there can be wet mounts that can be done. But those have a low sensitivity, so usually they don't do wet mounts, or they'll pair wet mounts with another diagnosis method. There's also the rapid test, which is the picture that I showed down below, which basically you do a swab and then you put it in the mix and then you let it sit, and then the results will appear either positive or negative. There's also the nucleic acid amplification testing. So those samples can be obtained using urine, vaginal, or endocervical specimens from men or women, or a urethral swab. And this one has high sensitivity, so it's used more often. Also, like I said, the sample of the vaginal fluid and then the swab as well. And side of the man's penis, and you can look at it under the microscope, and then you'll see, kind of like in this top picture here, you'll see the parasites floating around. So now for the T. vaginalis lab diagnosis. So it thrives at 37 degrees Celsius, and it prefers a slightly alkaline to neutral pH, which is about 6 to 6.8. It thrives in anaerobic conditions, so it will thrive with little to no oxygen present. And then the Diamond's medium is commonly used to culture this microbe. So there is an addition of small amounts of agar, which reduces oxygen tension, resulting into a more prolific growth of the trichomoniasis, which ultimately will grow and reproduce under anaerobic conditions. This microbe can also be pretty hard to culture in the lab as well, because it is sensitive to changes in the environment and it will require specific culture conditions. So now for the T. vaginalis virulence factors. So we have adhesions, specifically lipoglycans, that play a role in the adhesion of the parasite to the host cells, but they can also modulate the host immune response. And some other adhesions are adhesion protein 65 and P270. These also will help with the attachment of the parasite to the host epithelial cells. We have tetraspanins, which I'll talk a little bit more about in the pathogenesis. We have cysteine proteinases, which are enzymes that can degrade the host proteins, which ultimately contributes to the cytotoxic effects of the parasite, which causes damage to the host tissue and can promote inflammation. And then, like I mentioned earlier, they are capable of phagocytosing other microbes in the normal microbiota and can release lactic and acetic acid, which will lower the pH of the vaginal tissue. And the low pH of the vaginal secretion is cytotoxic to epithelial cells. So this is how the disruption of the normal microbiota starts. So now we're going to move into the pathogenesis. So the infective agent is the trophozoites. The incubation period is a pretty big range. There's not a definite incubation period about it, but they said anywhere between 3 to 28 days. So the use of lipoglycans to attach to the GAL1 protein. So GAL1 protein is a receptor for T. vaginalis that sits on the cervical epithelial cells. So this is the receptor protein. And so then in the picture here, the Trichomoniasis attachment to the epithelial cells is due to the binding of the parasite of the lipoglycan by GAL1. So the T. vaginalis attached to the epithelial cells of the urogenital tract, which is mediated by the interaction between the glycan domain of the TV lipoglycan and the GAL1. So the GAL1 is acting as the bridge between the host and the host cell and then the parasite surfaces as it's attaching, which is what is being shown in this picture here. So a little bit more about the pathogenesis. So the tetraspanins, which is an integral membrane protein, which influence the interaction between T. vaginalis and the host cells, the host immune cells, which is what's being shown in this picture here. So it allows the parasite to evade the immune detection or manipulate the host immune response to its advantage. So it can undergo antigenic variation, which will change the proteins on the surface, making it more challenging for the host cell to make a response, like we've learned in class. And then it can also release extracellular vesicles that induce an inflammatory response in the host cell with pro-inflammatory cytokines and chemokines will be released and then will cause a big inflammatory response. So in this picture here, for the first one that's on the left, so T. vaginalis infection can lead to the loss of the predominant lactobacillus species, which are the turquoise species in this picture here, which are very commonly found in the cervico-vaginal microbiome, and they promote increased vaginal colonization by a mixed and largely anaerobic bacterial population. And so on the bottom picture you can see that the diverse and largely anaerobic bacterial population that is part of the microbiome can help facilitate T. vaginalis colonization. So it is getting rid of some of the lactobacillus species in the microbiome, but T. vaginalis is easier. It allows T. vaginalis to attach to the host cells. And in the second picture here, the protective lactobacilli and the T. vaginalis are competitors in this microbiome. So you can see in the second picture how the parasite will inhibit the growth of lactobacilli, and then the bacteria inhibits the T. vaginalis adhesion to the epithelium, which causes a dysbiotic microbiota, which promotes T. vaginalis adhesion to the pathogenic biofilm in the host cell. So it enhances the host cell and epithelial damage. And once the microbiome is damaged, and it's hard to restore it, which is why you can see in the picture that some of the vaginal epithelial cells in the dysbiosis are grayed out because you can't really revert it. And then the modes of T. vaginalis. So some modes of it is a lot of damage. So damage to the host tissue from the parasite killing the host cell, disruption of steady state vaginal microbial ecology, and then activates the host immune response, which is that inflammatory response. So in this picture, you can see how the vaginal epithelial cells will attach, and then they will attach to other things, such as the macrophages, the B cells, T cells, and red blood cells that are all coming to the inflammatory site to help, but the T. vaginalis is attaching to all those other important cells and killing them. So the treatment is usually metronidazole or tinidazole, which is one large dose of medication that's only taken once usually, or it can be taken in smaller doses over a span of time, like an antibiotic, usually around 7 to 10 days. And then all sex partners need treatment at the same time, just in case someone else is an asymptomatic case. They might not realize they have it, but will still need the medication for it. It is important to avoid sex until the treatment is over and the symptoms are gone. And then, just like your antibiotics, you need to make sure that the full prescription is completed even after you start to feel better, to make sure that the infection is cleared completely, because it can come back. So prevention, risk factors, and complications. So prevention, the consistent and correct use of condoms during sex, regular STI screenings, or you can have abstinence. Some risk factors would be different in multiple sex partners, and then not wearing condoms during sex. And then some complications, if untreated. The infection could spread to the cervix, makes it easier for other STIs to enter the body. The increased risk of transmitting or acquiring HIV. And then associating the increase of cervical and prostate cancer, since the urogenital tract is where this microbe likes to hide. All right, and then my references.